So as Lei described, I've been a medical writer for over 20 years, 25 years probably, in pharma, big and small. Fortunately, I've had the opportunity to work heavily in clinical research on regulatory submissions, and also I've written protocols from the earliest stages of my medical writing career with the cross-functional clinical teams. And right now I'm a director at Janssen Pharma R&D, which is the pharma arm of Johnson & Johnson. I manage all the early development medical writing across TAs, all our therapeutic areas. But I have a focus on oncology and the immuno-oncology field, and is the big hot topic, as you probably know. So a little disclaimer here, these are this is my presentation. It doesn't necessarily reflect the opinions of my company. So the learning objectives for today are to understand the process for preparing a protocol for an initial IND or CTA for the EU. So the focus will be on first-in-human and proof-of-concept protocols for new clinical studies. So clinical pharmacology studies and operational aspects of setting up a study won't be covered today. So just a little survey. How many people here work in clinical research at a pharma company? Okay, a few. And, or how many work for contract research organizations or clinical operations aspects of clinical trials? Okay, a couple, all right. And how many are in academia or researchers or, okay, all right. All right, thanks. So in the planning and preparation stages for the protocol, it's important to look at the timeline and work with your project manager, especially if you're fortunate to have a good one like Lei Chuang. So the target is to get to that first patient, first dose, so enrolling that first patient in. So you try to plan backwards from that date that the team is targeting for. And also part of that planning involves planning around the submission of the IND and the clinical trial application. And so the accounting for the regulatory review timing of the submission, which includes the protocol, the investigator's brochure, all the non-clinical data, reports, summaries, in that initial IND application. Subsequent clinical trials don't have quite as extensive a package submitted, so you'll submit the protocol, maybe an updated IB, and some other background information, the informed consent form, and so forth. So the IND submission team is a cross-functional team that prepares and plans for the submission. The protocol writing team is a sub-team that focuses on the protocol, obviously. The clinician is the lead content owner of the protocol. There's usually a clinical scientist in my company as well, who's often a PhD person who works on the day-to-day aspects of designing the study. There's operations people, trials management people who manage setting up the logistical aspects of the study, the contracts with the sites, dealing with feasibility assessments of the sites to make sure that the study can actually be conducted at that site. The site has the capabilities of conducting the study, the right lab equipment and so forth, CT scanners available and so forth. Statistician's a very key person for the protocol, the pharmacokineticist, the pharmacodynamics and biomarkers person and translational medicine person, who may all be the same person, or maybe two people there. Biomarkers are exceptionally important now in oncology studies, especially with biologics and the immuno-oncology space. We're trying to get an early indicator of efficacy of the drug and what biomarkers the subject population has to study for future scientific purposes as well, to further define the population in later studies. And then the regulatory representative and of course the medical writer. Many companies have medical writers involved in protocol writing, some don't. So the early stage with the team, especially clinical, and the project managers, there's usually a compound development leader as well that manages all the development aspects of the drug. The project manager, like Lei, they'll put together high-level plans for the drug and get management approval. So they'll be involved in information gathering, researching approved treatments and those in development, the standard of care, the unmet need for that population, any non-clinical data that has been done on the drug or similar drugs, treatment guidelines. For instance, for oncology, in the US, there's the NCCN, which has treatment guidelines for physicians treating cancer patients. And in Europe, there's the European oncology treatment guidelines. And they're very detailed on how to use different drugs and what populations are applicable for certain drugs or treatments. And then, any health authority information, health authority approved treatments. Actually, the European website, the EMA, has what's called the EPAR area on their website, and they have a detailed assessment report on what went into approving the drug in the end, after all the studies were submitted and the summary documents and so forth were submitted and the drug was approved at the health authority. So it's actually publicly accessible. There's quite a lot of detail there on different drugs and what went into all the development and the approval of that drug in the EU. And the FDA also has a lot of information on their website as well. There's also published clinical studies, of course, and then registries like clinicaltrials.gov, where we're required to post our study designs and then, in the end, the study results for clinical studies. And at my company especially, we're very dedicated to transparency, data transparency. We're actually sharing data across other companies, and the EU has a big data transparency effort, so actually people can go and make requests for clinical data and clinical trials information. And then labeling and company websites have a bunch of information. I'm sure marketing at your various companies or other sources, consultants have a lot of information that are provided to clinical researchers for designing studies. different aspects related to phase 1 study designs. Again, my experience is oncology and biologics, some small molecules as well. So we look at getting the information to find that phase 2 dose as fast as possible. So the phase 1 studies now we're using what's called adaptive designs, where we're evaluating the dose-limiting toxicities in the population. We're trying to define the population as narrowly as possible. So we're doing some stratification on the populations we're enrolling as well. Since oncology, we're enrolling patients. I know some other therapeutic areas also enroll patients in earlier studies. In the initial phase 1, we're trying to get exploratory efficacy. But if you have an initial study in a therapeutic area where you've done healthy volunteer studies, this sort of assessment would be in more like a phase 1b type of study. So the adaptive design involves using flexible language on evaluating doses and regimens, but it needs to be prospectively designed and very carefully thought out. It's not totally flexible, but you're not listing out every single dose or regimen you're going to study. You say, the team or what we call the safety evaluation team will assess the safety and exploratory efficacy and PK and consider what the best dose regimens are in the study on a continual basis. So they'll meet frequently and look at all the data. So one thing we're doing is called accelerated titration and standard titration in these first-in-human protocols. So we're using this modified continual reassessment method based on a Bayesian logistic regression model with this EWOC principle. And it's somewhat involved. It requires a lot of support from your statistician and then your non-clinical group, your PK, your toxicologist, your what we call toxicokinetics. So there's a reference here where you can read more about that, all that terminology there. So basically, with accelerated titration, we're dosing in one subject at one dose level, and then 2 days later, we're dosing in the next subject at the next higher incremental dose level. And there's active safety evaluation, and there's DLT evaluation. There's a specific laboratory criteria for evaluating toxicities. So this involves very hands-on, active assessment of the patients. And there's extremely specific criteria in the protocols regarding this type of safety assessment and PK assessment of the subjects, even though you don't list out every dose, as I was saying. Standard titration then can be used if the safety evaluation team feels you need to maybe take a step back and expand a certain dose level with more subjects. And then we may explore different dose regimens in parallel. We may expand cohorts to include PK/PD, what we call PK/PD cohorts, to further explore different doses and dose regimens, to add more patients there, to explore various data, PK, even biomarker data. A more traditional design is a 3+3 design, multiple ascending dose design, where you enroll and dose 3 patients at a certain dose and regimen, and you evaluate the safety. And if things look good, you then go up to that next dose level. But if there is a concern, a potential concern or concern, then you would expand that dose to include an additional 3 subjects. And then you do an evaluation and see how many subjects had any dose-limiting toxicities or any adverse events that are a concern. And you decide if you can accelerate to the next dose or not, or you call that the maximum tolerated dose, or you can go down to the next lower dose level and expand that and see if that's tolerated. And you may call that next lower dose the maximum tolerated dose. So the goal of all this these new models is to get to that recommended phase 2 dose as quick as possible. We call it the RP2D. If you see that abbreviation, now you know what it is. Again, the starting dose considerations are very carefully considered, and we have a first-in-human review committee review things. We have, even before a first-in-human committee reviews the protocol and the investigator's brochure and the key toxicology study results, we have what's called a biologics committee that looks at all this data that we have as far as the no observed adverse effect level from the chronic GLP tox study. And then we'll often add in an additional safety factor. So say the no observed adverse effect level in monkeys, in the key monkey 5-week monkey study was say 5 milligrams per kilogram. We may add a safety factor of 5-fold to that, so go down 5-fold for the human dose. We also look at in vitro assays of the effect on the target molecule. We may have some human cell lines where we've done some experiments, and we may have receptor occupancy analysis, which looks at the PK and of the receptor, and it sort of involves modeling to get sort of a predicted dose for how much receptor occupancy there is. And this is more for monoclonal antibodies. And we're doing a lot of work with monoclonal antibodies in immuno-oncology, and also bispecific monoclonal antibodies, which is the next era coming up. And then we look at the predicted human PK based on animal studies, and then integrated that human PK with the tox data. So they do a toxicokinetics analysis. So then we propose the starting dose for the first-in-human study. So we'll say, we'll list the starting dose in the protocol, and then we'll say it will not exceed a certain level, but it doesn't rule out other doses and regimens might be explored, so we have some flexibility there as well. So with adaptive designs, there's been debate with the health authorities about them as well, because they are somewhat accelerated. So the FDA has a guidance. It's a draft guidance from 2010. The EMA came out with a couple things, a reflection paper in 2007, which was pretty high level, not very detailed. And just this past November, they have a draft guidance out for review on first-in-human and early clinical trials. It's a little bit more conservative, actually, and it involves doing amendments. So one thing with adaptive designs, we don't necessarily have to do a lot of protocol amendments to go to that next dose because we include the flexible wording with extremely careful and detailed safety assessments. One thing we're also doing is proof of concept studies. This is often done for combination drug studies where we have possibly a marketed drug together with a new drug looking at a very specific patient population, a heavily pre-treated population usually has a large unmet need. And we're looking at combination drugs, looking at the synergistic effects of each drug, in a lot of cases on the immune system. And the phase 2B aspect is called a safety run-in. Phase 1B aspect is called a safety run-in of the combination. So we want to, because we're combining these drugs for the first time in a patient population, even though we have phase 1 studies and one drug may be approved, the other drug may just have a phase 1 study that may be still ongoing. So we do the safety run-in. It's similar in some aspects to a phase 1 as far as the careful safety review, a DLT, dose-limiting toxicity evaluation, very careful criteria around that. But there may be only 6 patients in the safety run-in. And then there's an evaluation whether we can move to what we call the randomization phase, the phase 2 phase of the study. So it's a comparative study with 2 cohorts then in the randomized phase of the 2 drugs versus the one marketed drug, or maybe close to being marketed drug, maybe a drug that's been through phase 3 and has established efficacy and safety. There's some adaptive aspects to this as well. In the phase 1B, we also put in some wording about exploring other doses and regimens, because this is the first time we're comparing these drugs. So what we think might be the right dose and regimen may not actually be. So we're evaluating that, and we may expand with additional subjects in that phase 1B to explore different doses and regimens. And actually, the protocol could be amended to be a phase 3 registration study after an interim analysis and maybe some discussions with the health authorities like the FDA, getting scientific advice on that. I'm going to go over some of the background on doing our protocol, some of the regulatory guidance. So the International Council for Harmonization, E6, the Good Clinical Practice guideline has a large section on clinical protocols and amendments as well. So the audience for a protocol are the investigators of the study site research coordinators, the IRBs, and the ethics committees. Some are specific to a certain hospital or academic research center, or some are regional, especially in the EU. There may be some other special review committees for, like if you're using a recombinant drug, there may be some additional review committees. The FDA has what's called the RAC, the Recombinant Advisory Committee, and they actually review the protocol as well and other documents. And then auditors, health authority auditors, or internal company auditors are the audience. So it's important to be compliant in your protocol as well with regulatory practices and guidelines. And internal personnel use the protocol for various purposes. So the content of the protocol can be broken down to background, objectives, endpoints, instructions for evaluations, defining the study population, analyses, and then compliance aspects. And then there can also be supportive documents submitted with the protocol to the study sites and the health authorities. So you don't have to have every single detail in the protocol. You can have supportive documents like a lab manual that goes into the very detail of lab sample collection, like what tubes you're using and so forth. And then drug administration details. We include enough information in the protocol, obviously, but some detailed information on the administration sets, if it's an IV drug, or the syringes, if it's a subcutaneous drug, are left to another manual that's submitted with the protocol. So the initial section of the protocol is a synopsis. Usually it's intended to be a brief overview. We like to keep it to even like 2 pages, even for a complex study, because the clinicians and study coordinators at sites are very busy. They're doing maybe many different studies and have patients to see. So we try to make things as brief and concise, but yet complete and accurate and of good quality as well. So the synopsis provides an overview of the study design, and it's used for initial discussions with potential investigators and to gain initial input from health authorities before finalizing the full protocol. So we'll often draft the synopsis. Sometimes we call it an outline or a protocol elements document, and that can be used for these initial discussions before the actual full protocol is fully written. Then we include a study schema and a flow diagram of the study. The schedule of assessments tables. So we try to put all the procedures and timing and frequency in a tabular format. We may have a main one, and then one for PK and one for biomarkers, possibly. And, so this synopsis can be a mix of full sentences and bullets to keep things concise. So the next sections are the objectives, endpoints, hypothesis, and the assessments and stats in the what we call the outline or core elements, or the synopsis of the study. So getting to the full protocol, the introduction provides, this comes after the synopsis, and we put the time and event schedule, the schedule of assessments up front as well. So after that, we have the introduction, provides a background information and a good rationale for the study. We want to make it a very scientific basis for the study. So in an initial study, we summarize the non-clinical data that's available. So in a phase 1 protocol, this section would be larger and have more non-clinical information. Although we do refer to the investigator's brochure for the additional details in the protocol. We include any clinical data that's relevant if there was a phase 1 study done previously, and any review of the literature, applicable studies. That could be literature on similar drugs as well, or similar targets as well. So we really one of the main aspects of this is to have a good study rationale and goal for the study, and outlining the potential benefits and risks. The EU especially likes to have that up front. So we make sure we've defined the study population, any non-clinical data, any biomarker data that's been done, in vitro data that's been done to show that the drug could potentially have activity against the target, and that the safety profile looks to be reasonable from non-clinical studies. And then a dose and dose regimen justification, what I described earlier, all that non-clinical assessments that go into that dose regimen justification, and then a compliance statement that will conduct the study according to a good clinical practice and ethics, for conducting human trials. So that's an overview of the introduction. The objectives of the study, we usually have primary objectives and secondary, and sometimes we have exploratory objectives, which may relate more to the biomarkers or even patient-reported outcomes in early studies. Sometimes we include those questionnaires on patient quality of life in early studies to get a sampling of data using those patient-reported outcomes questionnaires. But the primary objectives for a phase 1 study are obviously safety, and then secondary can be preliminary efficacy, and or hypothesis generating as well. Exploratory objectives, the study won't be powered to really support these objectives, but it's more for gathering data for further scientific evaluation. And then each objective is written to have a corresponding endpoint, and the endpoints are what's exactly being measured in the study. So if you're measuring, if your objective is to determine the overall response rate in your population, the endpoint would be, using a standard evaluation criteria. For large molecules, for solid tumors, we use what's called RECIST, and IR RECIST, immune-related RECIST. It's a specific evaluation criteria for solid tumors, and it involves CT scans and measuring the tumors and various criteria for evaluating response. So you would say, using those criteria in the endpoint. So the overview of study design section explains the phase of the study, if it's randomized or blinded or open label, Open label means, everyone knows what the patient is getting, which is appropriate in an early phase study. You want to know what the patients are getting as far as the dose. So it describes the duration and parts of the study, this overview section, the enrollment, how enrollment is being done. Is it being done in a randomized way using an interactive web-based enrollment system, or how is enrollment being done? And the treatment, dosing, and then any follow-up procedures as well. At a high level, we give an overview of the study population, the number of subjects in the study, which the statistician usually provides that based on their power calculations. And then any stratification being done is for instance, is the subject, PDL or EGFR positive, based on genetic analysis. There may be some stratification done. And we try to define the arms of the study, or what we call the cohorts. So like I was describing in that comparative phase 1b/2 study, there would be a treatment arm with the 2 drugs, and then one with the single agent marketed drug. So you would define the arms and how randomization is being done. If it's being done 1-to-1, so if you have 80 subjects, 40 would be in each arm, or sometimes 2-to-1 is appropriate to get more data in the experimental arm, more subjects in that arm. And then we also provide fairly detailed rationales on the key aspects of the study, why we chose randomization, why we chose the response rate, for instance, as the primary objective, why we're doing PK analysis, what biomarker analysis we're doing, and why. And also, for instance, why we're doing other aspects like patient-reported outcomes. So we have paragraphs or 2 or 3 on the rationales, the reasons why we're collecting the data. So we have to have a scientific justification for all of that. So the study population is screened based on the inclusion-exclusion criteria listed in the protocol. The inclusion criteria, really defines that population. So we try to define the criteria for patients who have measurable disease and what that is, what that looks like. So we define that in the inclusion criteria, like that, genetic profile I was talking about, or have they been heavily pre-treated for multiple myeloma and failed, 3 lines of prior therapy. So we define all that very carefully, and maybe what therapies they failed. They usually should have failed standard treatment and get, have the opportunity to have had standard treatment before enrolling in this phase 1 experimental study. And then we have very detailed laboratory criteria for, especially for hematology, your anemia, platelets, and white blood cell criteria, liver enzyme criteria, very specific to ensure that the population is healthy enough to survive the duration of a clinical study. Then we have contraception wording, because people shouldn't get pregnant while they're in this type of clinical study. And then specific exclusion criteria is included. We want to exclude people who may have had other treatments recently. So we usually require them to have, a couple weeks or months, depending on the half-lives of the treatments they may have been previously exposed to. Or if they've been exposed to the same type of treatment, we might want to exclude them because they may have, failed an extremely similar drug, so they likely won't have benefit from this current study with our drug. And any other underlying medical complications we exclude as well to make sure that they're, they're healthy enough to undergo a clinical study. Then the different aspects of the protocol continue with the drug and drug administration. For phase 1, very specific management guidelines for toxicities. The immuno-oncology space, we're seeing, a lot of effects on the immune system. So we have very specific guidelines for treating patients or pre-treating patients with required concomitant medications, and any prohibited medications as well. And so these sections are getting larger and larger in our immuno-oncology studies with as we learn more about the different toxicities that we're seeing. So one of the main parts of the protocol are the actual study assessments that are being done in the study. So safety measurements, labs, ECGs, vital signs, of course. And then efficacy, there may be specific criteria or guidelines, like I said earlier, different scales, imaging, independent review may be involved of radiologic scan, for instance. Pharmacokinetics and pharmacodynamics biomarkers, what is being assessed from blood or tumor tissue to assess markers of the disease to help define the population for future studies or this study, and then markers of drug activity as well. We will include patient-reported outcomes, which are usually questionnaires the subject completes themselves. And in the study assessment section, we don't usually provide too much text on the timing frequency of assessments. We try, in protocol writing in general, we try not to be overly repetitive. Each section has its own purpose. We can cross-refer to other sections, to the table of assessments in the front of the protocol for the timing and frequency. So that makes, one thing just practically that makes when you amend the protocol to change certain things, it makes amending the protocol a little easier. If you don't have to change something in 10 different sections, you just change it in 1 or 2 sections and move on. And then one of the important aspects of a protocol after the study assessment section is the safety reporting. Safety reporting is highly regulated in clinical studies. We have to do expedited reporting of unexpected safety events, serious events. And we also do a severity assessment. Some protocols use a mild, moderate, severe assessment. Oncology studies and other studies actually use the National Cancer Institute toxicity grading system as well. So different events or laboratory events even are graded according to a scale of 1 to 5 for severity assessment. And the statistics section is an overview of the sample size calculations and the power of the study. The number of subjects is determined based on this power calculation. The sample size is created to have a high probability of detecting a true effect of a given magnitude. The outcome or endpoint is defined often in the stats section as well. Assumed values, what went into this power calculation, the proportion of subjects with an event, and then the type 1 error level, the power, and the sample size all are described in this section. So sometimes in phase 1 or for rare diseases, you may not have truly formal power calculations, but it's justified more on just practical criteria. A disease may be so rare that the health authorities will understand if you can only study 80 subjects in a phase 2 study, and that's all you can get in the world. Even if you opened 100 sites around the world. But you need a good scientific justification for that if you don't have a power calculation. And so the balance in clinical studies is to make sure you have a scientifically valid study without exposing too many subjects to an experimental treatment. So that's one reason why we're doing those adaptive trial designs to move from one subject to the next in an incremental dose escalation fashion to expose as few patients as possible to an experimental treatment where safety may not be proven yet, or efficacy either may not be proven. And then the stats section includes measures, analysis measures of how it will be summarized. Any interim analysis are described in detail in futility analyses. So there may be a cut of data at a certain point in time when a certain number of subjects have been enrolled and there's been a certain duration of treatment. And then there's an interim analysis done, and there's a decision made if the study is worthwhile to actually continue or not. But the extreme details and tables that will be produced are included in a separate document called the statistical analysis plan. And that's usually done after the protocol is done, before submission of a clinical study report. And you also need to submit it to the health authority as well to make sure you have a valid statistical analysis plan for the study. There's a bunch of administrative information in the protocol on data collection and management to ensure data monitoring and data quality is detailed. So we will list, is the data being monitored to the source data at the sites? So we'll actually send out people to the individual sites to look at any source data in the medical records and compare that to what data has been entered into the clinical database for the study to ensure data quality. And there are a bunch of regulatory and ethical considerations for the protocol. We actually write up, at least in my company, an ethical rationale for why we're conducting the study as a whole. What's the benefit risk to the patient? There's a description of the informed consent in general terms. It's what the patients sign to enter the study. So it provides an overview of the study and again, potential benefits and risks. That's a separate document, but it's described in the protocol. And it needs to align with what's in the protocol as well. We'll list a publication policy as well, that we are obligated to actually publish the results of the study in a peer-reviewed journal, and we will make that publicly available as well. Because we're doing, clinical research to benefit scientific knowledge of these patients and the use of the drug in these patients. So we need to make that knowledge public. And my company is quite a leader in that. We actually allow people to request clinical data through various means, and we actually partner with a program at Yale University that manages that, and a number of pharma companies are doing that as well. And also the EU we're working with them after we submit a clinical study report to the EU. We make that in a form that's can be publicly available, so the EU will post that after a health authority submission, a marketing application type of submission is done. So we have language around that in our protocols. We'll have a list of references to show that we did a thorough review of the literature in support of the study design. But no need to repeat extensive literature references listed in the IB, though. Just those main relevant references that you used. So this is sort of a high-level overview of the process for actually writing a protocol. As I indicated earlier, you want to do your homework on the study design. You want to gather all that information I listed earlier, background information on similar drugs, your own non-clinical research, and so forth, information on current study designs, will an adaptive design work or not? And then I like to encourage teams to have this, what I call a brainstorming session to discuss the details of the study design in a cross-functional team setting. I feel that one effective way of starting out a protocol, writing that early synopsis, is to have all the key functions together in a room, and or if you can't be together, we're very global nowadays, a WebEx with a video camera, if possible, to discuss and work through all the details, all the main details of the primary study design. Because as a medical writer coming in, I like to know that the team has thoughtfully reviewed and developed the study design. I don't mind being involved in those brainstorming sessions and organizing them as a medical writer, but I get value out of that, out of understanding what went into the development of the study design as well. And after 20 plus years of working in clinical research, I also can contribute things, even how to best write a study title, and I'll suggest some objectives even based on my experience. Then the synopsis is drafted, and that schedule of assessments, the timing. You want to get that dose and dose rationale down as early as possible as well. It's reviewed internally. I do like teams to get the key opinion leaders, the key investigators in that field, and potential investigators to take a look at the synopsis as soon as possible, because they're the ones out there who are going to conduct the study or may know of issues with certain aspects of the study design or the patients in the study. And then in the background, there's also discussion with regulators, health authorities. You may have a pre-IND meeting with the synopsis, as I was saying earlier, although we're not having as many of those anymore as we used to, because we can kind of predict what the FDA is going to say about a draft synopsis. And we want to, the team needs to validate the budget with the project manager as well. Make sure you have a realistic budget to conduct the study. So it's important to get the schedule of assessments done early so that you know what's being done in the study and then can evaluate costs as well. Then once that synopsis is validated, there may be an internal review committee looking at that as well. Then you're ready to draft the full protocol with the team. It usually takes about 2 or 3 reviews to get a really good draft together. The team may need to brainstorm some more as you're drafting the full protocol. And then you review with the team and other subsequent committees. We have what's called a PRC, a Protocol Review Committee, which includes members who are outside of that core team that are conducting the study and actually designing the protocol. So they are generally more senior people who have experience with protocols in that therapeutic area, but they're outside of that team. So they have sort of an unbiased perspective on the design and can add value to the quality of the protocol and the design. Again, like I said, for phase 1, there's also a first-in-human committee that reviews the protocol to make sure that all the thought went into the study design as far as is this protocol and drug really ready to be administered to humans. QC is an important aspect of document preparation. We make sure the document's internally consistent and is also technically ready to be submitted, because the health authorities have different technical requirements for electronic submissions as well. So then we have the team approve. We use a document management system at my company, and we do sort of a combination of electronic and paper signatures, and we archive all that approvals because we're a highly regulated industry and we can be audited by health authorities or our own internal auditors. So we make sure we have all this documentation that the content of the protocol has been approved and that we're compliant with the processes as well. So again, we have a document specialist also format the protocol and make submission ready, and then we submit it. So, yes. So I'm going to do a sort of an abbreviated exercise. I wanted to get through all this background material first. So I gave you some articles. The first one is, do you mind if I borrow this? So this first sheet here you have, I'd like you to kind of read through this text here on the front and first half of the back page, and try to write what you think would be the protocol title for this study, and also what would be the primary objective of the study as well. So a protocol title, you want to describe what phase the study is in. Is it a first in human, or phase 1, 2, or 3? It may be all of the above. So sometimes we get a little creative with protocol titles, but give your best shot at describing a protocol title. Some other aspects are whether it's randomized or open label. Is it a multi-center study? And then what's the population and any comparators or what drug is being studied? So read that first part. So I think we'll just do these first two, the protocol title and then the primary objective. Try to gain from reading what the main objective of the study is. Is it safety assessment of what, or efficacy, or both? And I think I'll skip this last part and save that for a workshop next year when hopefully I'll come back next year. So try drafting at your tables. You can work together at your tables. Try drafting the protocol title and the primary objective in about 10 minutes, within 10 minutes or so. Okay.
I'll present for our team that we have came up with the title. This is a randomized and two-arm combination study to evaluate. I can't read. Somebody want to read it out? Yeah. Nivolumab and with the ipilimumab versus nivolumab alone in patients with EGFR mutation positive NSCLC. That's the title. And the primary objective, should I continue or I'll
Sure.
stop here?
Yeah, go ahead.
Okay, primary objective will be to evaluate the response rate by the tumor size.
Okay, all right, great. Yeah, I think your title's very good. You probably didn't have enough information to know what phase it is, but it's likely a phase 2 type of study, so proof of concept study. So it may be a phase 1b/2, but it actually is a phase 2 study, just to let you know. And your objectives are also very good. Tumor response is the primary objective in that population, that defined population there, where they failed an EGFR line of therapy and another line of chemotherapy as well. Does anyone else want to share possibly what they wrote for an objective of the study? Or a title, either way.
The title is a randomized phase 2 study to compare the response of A plus B and A alone treatment in patients in advanced EGFR plus non-small cell lung cancer patients who have failed one-line target therapy and one-line chemotherapy. That's our title. And the primary endpoint we think is response rate.
Right. Okay. Yeah, I think that title is very good, very complete. I work with some physicians who can't write as good a title, so congratulations to both groups for writing great titles. And the objectives is to evaluate response rate. It can be, the objective can be fairly general like that, but you may want to say the response rate in these two cohorts, maybe to compare the response rate in these two cohorts. Maybe using a certain criteria, but you can just save more of the details of the criteria for the endpoint definition as well. But great job, thanks a lot. Does anyone have any questions actually about protocol writing? Or yeah, go ahead.
Study design is arm A patient will allow to cross over to arm B in the event of clear-cut disease progression. So what happened when we can't calculate their response rate? For arm B, will including this arm A subjects switch to the arm B?
Yeah, that's a good question. In these phase 1b/2 types of studies where you're evaluating this combination for the first time in a relatively pre-treated population, we often give the opportunity for the subjects in the single agent arm an opportunity to cross over to the treatment arm of the combination to get that benefit if they've had specific disease progression, if they've failed according to that specific criteria. So yes, I think there would be an analysis of both those subjects who've crossed over added into the primary combination arm as far as the overall response rate, but I think there would also be an analysis of just that combination arm response rate because they've had treatment for probably a longer duration by the time you do your primary analysis. So I think there would be a two-prong approach there as well.
So it really depends on when the crossover. So we will have one data cut point before the crossover. So we will be able to evaluate those different arms between a comparison and for the response rate. And also we evaluate the next cut point when they cross over, and it's another data cut point, and we analyze that. And also sometimes it depends, it's all about, that's why I'm saying the biostatistician they are so important because they will also look at the data we made. So it's really all about the post-studies evaluations after data cleaning and after database mark. And we will determine whether if we didn't see a very good signal, we will decide how to cut the data again and do another analysis trying to identify the subpopulations, the stratify the patients. So that's all different things, all the magic our biostatistician they are doing. And I think that if everybody are interested in biostatisticians, please, after today's symposium, please write the response, do the survey. We will send out a survey to everybody. And please respond to the survey what you are interested to hear next year. And we will invite speakers based on your response. Thanks.
Sure. And there'll also be a combined overall survival analysis too for everyone. Yeah.
Acceleration titration. You have this modified continuous reassessment, and this reassessment, if I'm understand correctly, it will be done every cohort, every single dose cohort, and what is the turnover rate of this assessment? Because this may add up the whole trial's time frame. Is that correct?
Yeah.
And so for the adaptive design, we want to reduce the total clinical development time. But will this affect the time frame of this trial?
Yeah. So the evaluation is done while the study is continuing. Once the predefined criteria is met and there's an interim analysis, they try to decide to get to that recommended phase 2 dose as quickly as possible once they start seeing markers of efficacy or tumor measurements, responses, in other words. And so in our early oncology studies, we're seeing some of these decisions happening within about 9 months of the first patient being enrolled in the study. So that seems quicker in my experience than the past studies of more traditional phase 1, 3+3, where you're waiting you're waiting over a year or two for that evaluation to be done.
I would like to know the publication policy for phase 1 study from this company and Ken for phase 1, because there are much more uncertainty for phase 1 study. So I want to know the policy.
Well, I don't have it in front of me right now, but I believe we're committed equally to publishing phase 1 results in peer-reviewed journals like we would do phase 2 and 3 with these patient studies. Healthy volunteers, maybe clin pharm studies may not have quite the same rigorous publication policy, but with our patient studies, we have the same policy for phase 1 as with phase 2 and 3. So we're at J&J, we're extremely committed to sharing information and making it available. Yeah.
I think the publication policy, there's no limitation for the publications. Basically, we finish the phase 1 studies and we want to publish the information as soon as we can, because it's publicity as well for from the company standpoint, because you want to let people know how quickly your molecule is moving. And so it's good for your investors as well. They want to see your molecule is moving and everybody knows that. There's 2 different publications. Something really moved really quickly is abstract. And so usually it depends on indication. You usually have a certain type of conference you will go to. And so usually once we have top-line data, we will quickly put a poster together, an abstract together to publish, to submit to the conference. We want to get the voice out to let people know what's our molecules, what kind of phase 1 data we have. And of course, we are not disclose all the details, because details, a lot of them, they still confidential. And then about a year after that, we will have we will have the data fully analyzed. Because at the time, abstract really publish the top line data. We call it top line data. Really is the first data, first cut. And you analyze data, you get to see the safety. For phase 1 is really the safety. And we want to say, you want to have something to say is, we test 22 patients, and our patient, the efficacy, the safety is, the drug is well tolerated. We won't say it's safe because we don't have enough evidence to say it's safe, but we say well tolerated. And we will say how many patients with grade 1, grade 1, grade 2, the AE, the grade 3, grade 4 SAE, how many patients they have, and to show they are well tolerated. And we will also say from the data we see the trend of such and such. So it's really well, this study we are going to pursue for the phase 2 study, something like that. And that's something in the abstract. And about a year later, you kind of, after database really completely locked, and your data cleaned, and you analyze everything, cut and dice everything, you'll be able to write a manuscript. And that's the second wave of the publications. And by that time, your phase 2 pretty much is on the way already. So that's phase 1 abstract data allow you to start prepare phase 2 studies or phase 1B POC studies.
Yeah, like Lei said, we share information at medical conferences and meetings through abstracts and posters and presentations very rapidly as well.
And I think I also want to say a lot of time when you have a patent, your method of use, a lot of time is
because you will claim you are using the data. Your drug is, if there's a first in class, and you have a composition patent, and you quickly file. You also file the method of use, and a lot of time you file a initiation of patent, and you actually 1 year later you subject to you file the patent. If you don't have data to back it off, the patent is empty shelf. It didn't really mean anything. So a lot of time is a year later you have to kind of start submit the data to support the patent. And so the same time when you start to have the data, your IP has to start working on when you do the publication. Before you submit the publication, that data has to be submitted to the patent office. Just make sure you do that, because that will be protected. And after that, you submit abstract. That doesn't matter.
Okay, thank you everyone for your attention, and enjoy lunch as well.
Thanks.
Thank you.